Water in which the reproduction is to take place is too rich in minerals, too hard, or the pH value is too low or too high, the eggs will usually die. Between the egg membrane and the yolk sac, with its embryonic blastoderm and blastodisc, there is a thin, hollow cavity. Water seeps through the embryonic sac and fills this space after the egg has been laid. This way, the contents of the egg can detach itself from the embryonic sac and move freely. In this process, an equalization of a difference in density between the aquarium water and the contents of the embryonic sac occurs, also called osmosis. If the surrounding water is too rich in minerals, the egg will shrink because water is being withdrawn from it. On the other hand, when the aquarium water contains too few minerals, the eggs will swell up and burst. Strictly speaking, the size of the tank is only of secondary importance. However, it should not be too small according to the size of the fish. Light should be kept somewhat subdued rather than too bright. Usually, substrate is not used, so that waste is easier to remove and the number of microbes can be kept at a low level. Decorational pieces, such as preserved roots that hang into the aquarium from above, need not be absent. These pieces, in addition to back and side glass panels in colors that are not too dark, give the discus a feeling of safety. For breeding, the temperature of the water should be approximately 30 degrees Celsius. It's also important the area around the aquarium be kept quiet. Unusual sudden movements are completely out of place at this time and should be avoided. And now to the breeding pair itself. You can tell by observation which fish harmonize with each other or leave the fish to choose their own mates. If there's only a single pair, rather than several males and females to choose from, the only thing to do is wait and hope. Despite optimal conditions, it may turn into a long trial of your patience, especially when dealing with fish caught in the wild. But what can be done in addition to this? We asked the experienced discus breeder Manfred Goebel, is it possible to force the discus to spawn?
Ich denke ja. Ich glaube, dass man das über zwei Wege.
I think so, yes. I think that there are two different ways to do it. It happens again and again in nature in exactly the same way. First of all, by means of the food. A change in food, especially when the fish are fed live food, such as black mosquito larvae, or other live food, such as white worms. So by changing the food, and secondly, a change in the water.
Also ein Wechsel im Futter und zum anderen eine Veränderung des Wassers. Genau wie es in der Natur auch passiert.
Just as it happens in nature, through the rainy season and through the dry season. Water changes cause the fish to spawn. The changes in the water do not have to be drastic. This can be achieved in the aquarium quite well with a normal water change using a good quality water.
Im Aquarium ganz gut durch einen normalen Wasserwechsel mit einem qualitativ guten Wasser erreichen. Man kann beim Discus durchaus nachhelfen, indem man
The discus can be assisted by installing a peat filtration. With these few changes, we have done all the things that will encourage the fish to spawn.
Was die Fische dazu bringt, zu laichen. Und das zeigt sich dann ganz schön in ihrem Verhalten.
And that becomes very clearly visible in their behavior. They will begin to swim in each other's direction, begin to look for their mate. They will begin to signal that they are ready to spawn with shaking and quivering movements. They will begin to look for an appropriate spawning substratum and begin cleaning the substratum. This activity is constantly interrupted by swimming in the partner's direction and palpitating. At this point, it can be observed that the caudal fins of the fish will become dark, and when the fish display the behavior natural to their species, they will also have light-colored or yellow throats during the entire period of spawning preparation. All this can be triggered by making changes in the food or by making changes in the water or, optimally, by making changes in both factors.
Discus fish belong to the group of guarding cichlids who deposit their spawn on a firm substratum. They prefer strongly slanted or vertical surfaces for this. Aside from sturdy leaves, a variety of shapes of objects are gladly and readily accepted as a spawning substratum. For example, clay pots turned upside down with predominantly slanted surfaces. Even baked bricks with only vertical surfaces are used and accepted. Sometimes the rough clay surface becomes covered with algae because there's too much light and is difficult to clean. Therefore, a plastic tube with a sufficient diameter and a fine surface structure can be placed in an upright position for use as a spawning substratum. Sometimes the discus will also spawn on the vertical side panels of the tank, but very rarely on the horizontal bottom pane. So much for the preparations for successful breeding. Are there other factors that have to be taken into consideration? We asked successful discus breeder Jürgen Luckner.
Ich möchte noch einige Anmerkungen zur Zucht bzw. deren Vorbereitung machen.
I would like to make a few comments about breeding, or rather about the preparations. I am an advocate of good, healthy aquarium water. This means water that is biologically intact. If this kind of water is not available and you are dependent on partially desalinated osmosis or spring or tap water, it should only be used after being run through a well-functioning filter for a while and being enriched with minerals and trace elements. Before the breeding fish, or rather the breeding pairs placed in the aquarium, prepared for them, make sure the animals are free of parasites. If this isn't the case, they should be treated with the appropriate medication. If this doesn't take place and the parent fish do have parasites, such as gillworms, these can be passed on to the fry and result in the loss of part of or even the entire spawn.
The laying of the eggs usually occurs at midday or in the late afternoon on a firm substratum the parent fish have previously cleaned with their mouths. Apparently, the parent fish determine when the embryos will hatch based on water temperature. The developmental period of up till the time of hatching is between 55 and 65 hours, depending on water temperature and water values. This means the embryos of the eggs fertilized at midday and in higher water temperatures will hatch on the evening of the second day before nightfall. When fertilization has taken place in the late afternoon and by lower water temperatures, they'll hatch after nightfall on the morning of the third day. Hatching at night in the dark would be lethal for the larvae, at least in the wild, because the parents would not be able to gather them. Adult discus fish usually lay a large number of eggs, often as many as 200. The eggs are approximately 1.4 millimeters in length and have a diameter of nearly 1.2 millimeters. Just as in the case of the terrestrial globe, an egg has two opposite poles and an equator. At the upper pole, we find the opening of the egg membrane, a funnel-shaped invagination of the egg membrane that is important for fertilization. Through it, the egg is fertilized by the spermatozoon. Like many other cichlid eggs, the eggs of the discus adhere to the substratum in a longitudinal position. A very thin layer of mucus forms along the egg equator, firmly adhering the egg to the surface under it. Reproduction is started with the laying of the eggs. The female swims from the bottom to the top, just above the surface of the substratum, and successfully lays the eggs. They are fertilized immediately by the male, who also swims just over the surface of the eggs, from the bottom to the top. During this process, there should be no strong water movement in the aquarium. For example, an inflow back into the aquarium from the filtering unit. The female lays the eggs from the bottom to the top, almost only in one direction, so that the opening of the egg membrane is pointing upward. Normally, the male fertilizes the spawn after each egg is laid. To fertilize the egg, the sperm must try to reach the funneled opening of the egg membrane located on the upper pole of the egg. Because the spermatozoan can usually only survive for about 30 seconds, they must reach the egg within this short time. Strong water movement would make this uncertain. Reproduction first begins with the entrance of the spermatozoan into the egg cell. On average, spawning takes 70 minutes. Afterwards, the parent fish begin caring for the eggs. They stay near the nest, and usually taking turns, they direct a constant, weak flow of water toward the eggs with their pectoral fins. Firstly, this provides a good supply of oxygen-rich water, and secondly, it prevents dirt particles and microorganisms from collecting on the nest and harming it. The parent fish observe their nest very attentively. During this time, and in the days that follow, a faint night lighting above the nest is highly recommended for a better orientation for the parent fish. After successful fertilization, the embryonic blastoderm, which lies under the opening of the egg membrane, begins to develop, and cell division begins. In an advanced stage, this is called the cleavage of the ovum. In the polycellular stage, the blastodisc lifts itself up from the yolk and remains connected to it only on the edges. The resulting space underneath is called the cleavage cavity. Until this time, the blastodisc is still completely smooth. By looking at it, you can only guess where the head or the tail of the embryo will develop. The healthy yolk, marbled with fat particles, is clearly visible. The required oxygen is absorbed from the surrounding water by way of the embryonic sac. While the blastodermic layer develops inside, the development of the embryonic tissue, which slowly grows over the yolk from the front upper portion, is clearly visible through the embryonic sac. All of these processes are perceived by the parent fish caring for their eggs as changes. In these few hours, nature is in quite a hurry. 26 hours after fertilization, it's apparent if the environment of the eggs, that is the water temperature and so on, will allow for the development of new and healthy life. If this is not the case, many of the fertilized eggs will die off. Their yolks will curdle, and the accumulation of cells, or rather the blastodisc, will dissolve. Their life is over, after having barely begun. At this time, a healthy, normally developing egg will show very distinct changes. Approximately 40 hours after fertilization, the first fragile signs of life become visible. Embryonic fluid is pumped through the developing body. The first signs of the developing embryo begin to appear with the swelling of the head and the embryonic spine, which will later become the tail. The contents of the embryonic sac will become noticeably darker. The parent fish now watch the development of the eggs very attentively.